DNA replication. DNA replication is the process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. Replication is an essential process because whenever a cell divides, the two new daughter cells must contain the same genetic information or DNA as the parent cell.
Lagging strand. A lagging strand is one of two strands of DNA found at the replication fork or junction in the double helix. The other strand is called the leading strand. A lagging strand requires a slight delay before undergoing replication, and it must undergo replication discontinuously in small fragments. DNA polymerases are enzymes that create DNA molecules by assembling nucleotides. These enzymes are essential to DNA replication and usually work in pairs to create two identical DNA strands from one original DNA molecule. DNA ligase is an enzyme which can connect two strands of DNA together by forming a bond between the phosphate group of one strand and the deoxyribose group of another. It is used in cells to join together at Okazaki fragments, which are formed on the lagging strand during DNA replication. RNA primer. A primer is a short nucleic acid sequence that provides a starting point for DNA synthesis. In living organisms, primers are short strands of RNA. A primer must be synthesized by an enzyme called primase, which is a type of RNA polymerase before DNA replication can occur. DNA primase is an enzyme involved in the replication of DNA and is a type of RNA polymerase. Primase catalyzes the synthesis of short RNA segments called the primer complementary of single-stranded DNA.
DNA polymerase removes RNA primer and fills in the nucleotides which are necessary for DNA formed in direction 5'3". Single-strand binding. DNA binds proteins in DNA replication, recombination, and repair. Helicase. Helicase are enzymes that bind and may even remodel nucleic acid or nucleic acid complexes.
Topoisomerases are enzymes that participate in the overwinding or underwinding of DNA. Okazaki fragments are short sequences of DNA nucleotides which are synthesized discontinuously and later linked together by the enzyme DNA ligase to create the lagging strand during DNA replication. The replication fork is a very active area where DNA replication takes place. It is created when DNA helicase unwinds the double helix structure of the DNA. The replication fork looks like a fork in the road that is composed of a leading strand and a lagging strand of DNA.
Double-stranded DNA consists of two polynucleotides chains whose nitrogenous bases are connected by hydrogen bonds. With this arrangement, each strand mirrors the other as a result of the anti-parallel orientation of the sugar phosphate backbones, as well as the complementary nature of the A to U and C to G pairing. Phosphodiester bonds is the linkage between the 3-carbon atom of the sugar molecule and the 5-carbon atom of another, deoxyribose in DNA and ribose in RNA. Strong covalent bonds form between the phosphate group of two 5-carbon ring carbohydrates over two ester bonds.